Should we start? Now, some of you should just put this on the group that after 10 minutes of that nobody will be taken in, है ना? तो ये 10 मिनट के अंदर सब लोग आ जाएंगे तो it will be easy. Then will not take the person inside. Okay? Should we start? Okay. Now, see, we'll proceed further. We have seen up to diuretics, and I have given you the assignments. Similarly, you have to go for all the topics. Okay. Now, basically, I'd like to add a few things before you go for that. What is very important is whenever we are going for this, we'll find various kinds of models as we have seen. We have seen in vivo model, in vitro model, ex vivo. So these models are actually what they are making the condition most, resembling to the disease conditions. Okay. And these disease conditions are there that we have to produce. So in each case, you'll find that is one thing. Second thing is, we have to understand what is the cause of the disease, what is affecting the person, or what is, because we know pharmacology also. Example, for Alzheimer's treatment, what are the drugs which are used for Alzheimer's? Anybody? Name the drugs which are used for Alzheimer's disease.
Donepezil, sir.
Donepezil, very good. Any other?
Rivastigmine.
Very good, very good. Okay. Can anybody tell what is the mechanism of action of this drug?
Sir, they cross the blood-brain barrier.
Very good, very good. So, is it enough to cross blood-brain barrier?
Lipid soluble.
Okay, they are lipid soluble. Any other thing?
No idea.
Very good, no problem. Anyone can tell what is the mechanism of action of either donepezil or rivastigmine? No one. Yes. It inhibits the hydrolysis. Very good, Aarti. How it inhibits?
acetylcholinesterase inhibit करते enzyme.
Very good. It inhibits the acetylcholinesterase enzyme. Now, what is our knowledge now? What knowledge it gives? इससे तुमको क्या नॉलेज मिला? इफ एनी ड्रग विच कैन इनहिबिट, देर आर टू थिंग्स विच वी आर टोल्ड नाउ, है ना, आज दो चीजें बताई मेरे को. मेरे क्वेश्चंस थे, डू यू नो एनी ड्रग विच इज यूज इन अल्जाइमर्स? दैट वाज करेक्टली टोल्ड, डोनेपिजिलन, रिवास्टिगमिन एटसेट्रा. देन माय सेकंड क्वेश्चन वाज मैकेनिज्म ऑफ एक्शन. उसको एक ने जवाब दिया कि दे क्रॉसेस ब्लड ब्रेन बैरियर. ओके. और दूसरा एक जवाब आया कि दे इनहिबिट द हाइड्रोलिसिस ऑफ एसिटाइल कोलिन. तीसरा जवाब आया कि दे कॉजेस द इनहिबिशन ऑफ एसिटाइल कोलिन एस्टरेज एंजाइम विच इज causing hydrolysis of acetylcholine, and because of that they are causing increase in the acetylcholine levels, है ना? Is it correct?
Yes, sir.
Is it correct?
Yes, sir.
Okay. Now that means that the drugs which are increasing the acetylcholine concentration in the particular area of brain, okay, they are increasing the memory, because the memory was lost in the Alzheimer's disease. So they are improving the memory. That means that any drug which can inhibit the acetylcholine esterase enzyme and that can cross blood brain barrier, because it is very important that drug should cross blood brain barrier. So when the drug is having two categories, then the drug can be a good candidate as a memory enhancer. To this type of drug we call them as a nootropics. Nootropics means which causes increase in the memory. Okay. So this is a one way of mechanism. Second mechanism can be what? There are certain drugs which improves the blood flow, therefore they improve the supply of nutrients, that is oxygen and glucose, and thus they improve in the memory function of this. Okay, so there are variety of mechanism by which they are acting, and accordingly we can study it. So can we study it in vitro? Yes, because if we can get the enzyme acetylcholine esterase enzyme in vitro, we can see that whether the drug is able to bind this acetylcholine esterase enzyme. If it is able to bind and it will prevent the acetylcholine metabolism or hydrolysis of acetylcholine, then yes, we can say that it is doing it. So these are the models which are based on various cholinesterase enzyme, and we are studying at various level. Okay, so these are the various models where we are studying the in vitro inhibition of acetylcholine esterase enzyme. It can be done in rat striatum. Similar other enzyme is there, that is butyrylcholinesterase act. So we can do it in human serum because it is present in the human serum. We have ex vivo cholinesterase inhibition activity, then molecular form of cholinesterase form rat frontal cortex and striatum. Basically mechanism is same in all cases. We are not, it's not a very big thing we are doing. Basic thing is we are trying to see the action, whether the drug can block acetylcholine esterase enzyme, whether it is butyrylcholinesterase or acetylcholinesterase, and accordingly we are trying to say that, and if the drug can cross blood brain barrier, so we have got so many actions which can be showing it. Okay. Then there can be drugs which can cause or which can increase the release of acetylcholine from the synapse. So we can check about these drugs. Then there are some drugs like oxytremorin, which is able to bind the muscarinic cholinergic receptors, and it is acting as an agonist. So we can see again with this radioactive isotope we can have, and we can see that whether this drug is able to bind to the muscarinic receptors and then we can analyze. This is again in vitro type of studies. Then stimulation of phosphatidyl inositol, that is IP3 we call, that is inositol triphosphate, and we can see that whether what is happening, because when acetylcholine is acting, it increases the, if you remember, IP3 DAG pathway. So it improves the IP3 production, and we can see that whether it is doing or not. So we can, this is a parameter, measurement of IP3, that is inositol triphosphate. Then again the radioactive isotope, that is 3H, hydrogen otherwise having only one, deuterium is having two, and this is 3H, that is the isotope which is a radioactive isotope, and we can see that whether it can bind it to not. Then we can have cultured neuron or astroglial cells, and what is, whether our drugs can improve the activities of that cultured neurons or that astroglial cells, that we can see. So these are various mechanisms by which we can have in vitro test, and there are in vivo tests are there, which are, if you remember earlier you have seen, there is a one equipment, we call it as a pole climbing apparatus. Anybody knows about pole climbing apparatus?
No, sir.
ओके तो एक काम करना, इसका भी यूट्यूब वीडियो देख लेना, पोल क्लाइंबिंग. I'll just tell you what is basic in that. In video you will understand in better way, because you will find the apparatus also. There is a box shape type cupboard type structure in which one pole is there, and surrounding it there are the mesh is there, steel mesh is there, and this is having variety of modifications. One is what, that when there will be buzzer, after buzzer immediately the electric current will be passed through the mesh, so that rat will sense the shock. If the rat has got good memory, then what rat will do, as soon as buzzer is there, it will climb on the wooden pole. तो वो पोल पे क्लाइम कर लेगा, पोल पे क्लाइम करेगा तो उसको शॉक नहीं लगेगा. ठीक है. सो दिस टाइप ऑफ एक्टिविटीज दे आर डन फॉर चेकिंग द मेमोरीज. You understood or not?
Yes, sir.
That is called as a pole climbing. फिर there are various other models where these are called as a what we can say, various maze. maze मालूम है maze किसको बोलते हैं? हाँ दानेश्वर बोलो. डू यू नो मेज?
सर मेज apparatus पता है सर, प्लस मेज वाला.
वेरी गुड, वेरी गुड, बहुत अच्छा. हाँ प्रतीक सूर्य ने बहुत अच्छा बोला, भूल भुलैया. So maze is a bhul bhulaiya. Very good, Pratik. So this is a maze is there, and in maze they have got variety of this thing, that there are two basic things are used for that. One is called as a reward system, and another is a punishment system. That is, if there are in maze there are variety of mazes are there, plus maze, elevated plus maze, then 8-arm maze, like that. There are variety of such maze are there. Then water maze is there. Basically all these mazes, they give some hint to that animal, small animal. These are all in vivo methods. That is, whole animal has to be used. So in that maze, when the animal goes, in one place there will be some food material will be there. The animal will be kept hungry, and the food material will be there, and some indications will be given for that. So if the animal can rightly go to that particular place, that means the memory is good. If it cannot go to that particular place, then that means memory is impaired. Now, apart from that, simultaneously we have got variety of models, either chemically induced or surgically induced, that we specifically destroy some of the area, okay, and or we can destroy it by producing hypoxia to various cells of brain. So there are a variety of ways by which we are affecting brain function. So this is one part of the model, and other part of model is the instrumentation. That instrumentation will allow the animal to reflect the memory status. If the animal can remember that where food is there and there it will go, that means good memory, as I told you, because it will be marked accordingly. Or where the animal gets punishment, there it goes, that means it's not a good memory, it is going towards a punishment section instead of going towards a reward section. And that is how we judge the memory, and on that basis we go for variety of this, okay. On that basis we can have variety of activities. So these are all various activities. Basically how these are done, that is, as I told you, active avoidance, that means once there is a buzzer, before the shock it will go away, okay. Then there is a maze which called as a water maze. The animal will put into a bucket, and there will be one place where some wooden block is given to animal, okay. That block will be almost below water again, but in particular area, animal can remember that in which area the block is there, so that it can rest on that. So these are various areas, and on that basis you can have a variety of such models which will able to screen the activity to what we call as a nootropics. Nootropics means memory enhancers, the drugs which can enhance the memory. So these are a variety of models we have got, and as I told you, what are things which are actually involved into it. One is what? Basically use of various instrument or various method, particularly for testing. So it can be the pole climbing apparatus, it can be the maze. Maze again, it can be different type of maze, it can be a shuttle box model, it can be a spatial. Here the word is coming, see here word is there, spatial, okay. It is not spatial. Spatial means to understand the location in which area it is there. That means when we say about maze, that means which area is rewarding, which area is punishing, and on the basis of slight modification or some color indications or some structural slight modification, the animal understands and remembers that this is what the rewarding position and this is what the punishment position. So when the animal goes only towards a reward position and it does not go towards a punishment position, this is what we call it as a spatial habituation and learning, or spatial discrimination. That is, it can identify that which is a rewarding place, which is not rewarding, or which is punishment place accordingly. So we have got again various mazes for that. Then visual discriminations on the basis of colors or on the basis of change in the appearances. Then spatial learning in the water maze, as I told you. Then next is olfactory learning. Yes, olfactory means smell. On the basis of smell, again some smell will be there in that particular area, and that area will be having either punishment area or rewarding area. And if the rat identifies this smell means punishment, this smell means reward as a food, then the animal will accordingly do it. There can be again many things in pole climbing and all. You will find that there will be some knobs will be given, and this knob will be showing that on that basis, if that is pressed, then they will get rewards. And if to study it again in higher way, if it is pressed repeatedly or at certain period in between, there will be maybe suppose first time reward or first 2 times there will be shock, and third time it will be giving reward. So again at that time, those which are having good memory or those which are gaining good memory, what they will do is after pushing immediately they will run away towards a non-shock zone, and then third time they will go for reward. So basically this is how they are going for, and accordingly the animals, they are either normal animals as I told you, then either they can be animals which are surgically or chemically, where amnesia is induced surgically, chemically or experimentally. So we are inducing experimentally amnesia. Amnesia is a forgetfulness. So we can induce that amnesia, and accordingly we can use various kind of situations. So please go through this. These are a variety of models which are available, and wherever you do not understand, we will discuss this. Okay. In all cases, again in future, what it will be the condition, let me tell you, for all kind of disease, once you understand the mechanism of disease or mechanism of the drug which are acting for the diseases, whatsoever diseases it may be, then it gives you idea that yes, that can be the way by which new drug can be studied, because if the one drug is active through that particular mechanism, then the next drug should also be active. So this is how the drugs are particularly evolved, or new drugs are coming. Or sometimes they identify targets first, that is, if this is there, this is the target, this is contributing into the pathophysiology of that particular disease. Now if we can arrest it, we can block it, then we can reduce the disease conditions, and on that basis our drugs can be active. So once we understand the basic causative things, okay, the either it is biochemical changes, genetic changes, structural or anatomical changes, etc., then we understand that why there is a disease. And second thing is, when we understand that how we have rectified it, suppose just like we have seen earlier that we can have various drugs which are acting through one or other mechanisms. Hypertension example we have taken. So we can have calcium channel blockers. So we know that any drug which is having calcium channel blocker activity, it will acting, or it will be helping as, it will be working as a antihypertensive. Then ARBs, that is angiotensin receptor blocker, ACE inhibitors, then beta blockers, and we so many. So we have got so many ways, so many mechanisms by which one can control hypertension. We can bring back the hypertension to the normal one. So all these methods, they can be available as a in vivo or in vitro. So that we can study, that one can check with. Those who have studied the differences between all, as I told you, each one is having their own advantage and own disadvantages. When you go for in vivo studies, you get a clear picture that whether any other additional activity the drug is showing, whether in addition to, if you remember drugs like chlorpromazine, anybody knows chlorpromazine or typical antipsychotics? Yes, chlorpromazine has got so many effects. Okay, it can having alpha receptor blocking effect. It can has choline receptor blocking, apart from the dopamine receptor blocking activity, which is there, which is used as a antipsychotic. So similarly, once you are using them in vivo, then you will able to get, yes, but apart from that, Arthi, other impacts are also there. Blockade at D2 receptor, plus it is having antimuscarinic effect, it is having alpha blocking effects. So because of all these multiple effects, we can have so many adverse effects, and it can be seen. So similarly, when it is in vivo, you can get all these multiple activities done, or multiple activities simultaneously observed, is one thing. Second thing is what? You know actually what is the impact, because there are so many things which are working when it is an in vivo system. There is homeostatic functions, there is a feedback mechanisms, there are immune mechanism, everything is working when it is a live system, in vivo system, and therefore in vivo gives you that clear picture. But many times you may not be able to very clear in the mechanism of action, because the drug is causing decrease in the blood pressure, but you may not be sure whether it is having alpha blocking, whether it is having beta blocking, whether it is having ARB blocking, or something else. So in that condition, when it is in vitro activity, whether it is on tissue or whether it is on cell lines or enzymes or something like that, then you can very clearly pinpoint that this is the action which is being understood or observed. So whatsoever drug you have studied as an antihypertensive using in vivo technique, now in vitro technique will help you to understand basic mechanism of that particular action. Okay, so both are complementary to each other, both are important, and therefore it is very important. So we'll understand various activity, and on that basis we can have the understanding of various drugs. So please go through this topic, that is what are various drugs, how we are testing it, and if we understand it, you can again go into a tabular form. Same thing for this also. What are the in vivo and what are the in vitro studies, how the models are there. We can just rush through various models which are being used. Okay, there are so many models which are there, as I told you, in vitro. So again, here we are measuring the acetylcholinesterase activity in rat stratum. Okay, so what we are doing is we are taking various reagents in which these buffers etc, and the drug we will be adding. We are getting the brains from that. The stratum is nigrostratum portion will be, or corpora strata is dissected, and it is ways homonize. The machines are there, we call that is called as homonizers, and then in that these cells will be there, and enzyme activity in the brain will be measured using the spectrophotometer, because if these enzymes are active, the cell are good, cell are viable, then enzymes will be more, and accordingly the activity will be seen more. Okay, so this is how we are getting, and accordingly we will understand what is the activity. Similarly, butyrylcholinesterase enzyme, which is present in serum, human serum, from that we can get it. Again, various chemicals on that basis. These are the reagents are there. Like buffer, usually we need in very high amount, because we need to maintain the pH during that particular situation. Basic mechanism is same, that is using this dithiobis nitrobenzoic acid, that is called as a DTNB, and this DTNB will show the changes or the enzyme activity. Okay, so this is how all these activities are being tested. Then next is in vivo activity, cholinesterase activities, In vivo, we need certain machine for that. Okay, so there are we can do with the machine. What we will do is 4 male star rats will be dosed intraperitoneally or orally, and rats are given varying doses of drug and sacrifice after 30 minutes or 1 hour after dosing, and we'll see the animals are observed and the occurrence of cholinergic sign if noted, that is pilorection, tremors, convulsions, salivation, diarrhea, and chromatoacroria. So these are the things which can be observed. Pilorection is tail is elevated. In body, tremors are there because of the acetylcholine. Acetylcholine can cause tremor, convulsions also. It will increase the salivation in the animals, can cause diarrhea. Okay, and another is this so these are sort of tears which are coming from the rats. Okay, so this is the presence of pigment in the normal tears of the rat. So orangish color liquid will be there around that, and that will be called as this chromodacronia. So this is various ways by which you can do it. Okay, then male star rat decapacitated, that means their heads are removed, heads are separated from the body, brains are rapidly removed, and the corpora striata, which is the area which is known for memory, that is removed, weighed and homogenized. The basic procedure will be same everywhere whenever homogenizer is there. Some scientists will just change the buffer. Some will use phosphate, some will use citrate buffer, so that doesn't make much difference. That particular pH will be maintained, and accordingly these buffers will be used, and these homogenates are used for the assays, various assays, and that is called as a percent inhibition, that is measured. That is how much color is absorption is there spectrophotometrically. So this is how it is done. Example of one table is given here, physostigmine when it is given. Okay, that is after 15 minutes, that is 0.25 hours, the percent inhibition is 46%, and the signs are pilorection, tremors, and diarrhea were found. After 0.5 minutes, it was percent inhibition was 28.5%. Okay, and all these three signs were found. After 1 hour, diarrhea was not there. After 2 hours, again diarrhea was not there, and slowly, gradually, the action was decreased. So immediately in 15 minutes, the maximum action is seen, which gradually decreases after intraperitoneal increases. So these are how various models are developed. Then molecular forms of acetylcholinesterase from rat frontal cortex and the striatum. So these again, which we can remove from this striatum, okay, frontal striatum. We can isolate it, we can, and then we are using the enzyme assays or enzyme studies. So on that basis, we'll see the enzyme kinetics, that is, if acetylcholine is given, how fast it is metabolized, and what is the impact after the acetylcholinesterase inhibitor drug. If any drug is having acetylcholinesterase enzyme inhibiting activity, then what we will be finding. So this is how we are going for the measurements of various activities. So activities are like preparation of isolation of molecular format of acetylcholinesterases, assess for the marker enzymes which are giving the idea about it, and enzyme inhibition studies using this enzyme, that whether the enzymes cholinesterase enzyme and other enzymes are inhibited. So this is how we get, and if there is a good IC value, that is inhibitory concentration value, then we can say that this drug is having activity. Which activity? Acetylcholinesterase activity. And if the drug is having acetylcholinesterase activity, that means the drug can be a nootropic in nature. Okay, this is radioactive, as I told you. This is a radioactivity. 3H means radioactivity. One of the hydrogen will be normal hydrogen will be replaced with this radioactive isotope, and then we can see the binding with these receptors. Okay, we can measure this techniques. You will see that how this binding is there, radioactive isotope has bound, and where it is causing any interaction with any of the neurotransmitter. So this will be the activity which you can go for, which you can study. So what you will do in that case is this: you have to synthesize having 3H, okay, instead of the normal. And this is, as I told you, it is a nuclear medicine, or it is a medicine with radioactive activity. For that, if anybody has to conduct it, a special permission has to be taken for that. Okay, the tissues will be there, and accordingly assay will be made. Tissue is removed. Same procedure as we have seen earlier. Rats are decapacitated, brains are removed, and accordingly the slices are prepared. Either you prepare slices, or in some cases homogenates are prepared. That is, there is a machine that will crush the tissue. Okay, and slices are there. They are placed on cold oxygenated buffer and incubated at 35 degrees Celsius for 30 minutes under oxygen. So this buffer is decanted, and the slices are placed on the nylon mesh in the stimulation chamber, and then you measure the activity on this machine to see that whether the drugs are able to bind with this. Okay, so this is how conditions are for it. You need not to remember this special condition which you require for that. Only what basic mechanism is there, how drugs are there, what are the models, and what are the parameters, and what is the brief procedure. This much is there. No need for the special care to be taken, etc., because that much depth is not expected. Okay, so this is how we are going for that. That is this is computerized program. That is this is old one. Lotus was the old one. Now we have got so many other softwares, so many machines, because I told you this book is very old one. Even second edition is almost 10 years back it has come. So this is done. That time Lotus was there. Now there are many other good softwares are available, and on that basis you can measure the changes which are occurring over there. Okay, so this is how various studies are there. Again, you will find that procedure is almost same in all places. Only slight modification will be there in basic what we can say activity measurement. So you can go through this, and if you have any difficulty we can discuss it. Okay, so you can make the table. Hello.
Yes, sir. Yes.
You can make the table of it. Almost you will again find, only thing is everywhere that this is the basic thing, that is this 3H, that is the radioactive isotope. Only thing is now it is put into oxytremorin. Earlier it was put into norepinephrine. Okay, see. So what is the will happen. Then earlier it was there in the same way, it was there with the 3H acetylcholine. So what is done is, to check this, we will give, what is the precursor of acetylcholine?
acetyl-CoA.
Acetyl-CoA and choline, isn't it?
Yes.
Two things are there. So we will incorporate this 3H into it, okay, and accordingly this is the choline chloride, that is the choline analog. So this choline analog we will be giving, and this choline analog will be put into animals there, so that whenever acetylcholine will be synthesized, that will be radioactive one, and we can see that what is happening to the release pattern of that. If its release increases, that means more choline is there, more acetylcholine is released, that means drug is neurotropic, and if it is decreasing, that means it is not a neurotropic drug. So this is how. Then here binding sites it will be studied, oxytremorin. So whether it can bind to that, how it is binding. Similarly, you can modify the structure. If it is a new drug you are able to synthesize, so you can again put this 3H, and you can say that will try on the muscarinic receptor. If your drug is also binding with the muscarinic receptor, that means the your drug is having nootropic activities. Okay. Then again this is radioisotopic methylscopolamine binding in the presence and absence of this GPP, that is G protein linked, this amine sequence is there. So that is known as a guanyl amidophosphate, that is GPP. Okay, guanyl amidophosphate is there, and here 5' is there. So this GPP is NH is the amine group, okay, and this is a phosphate. So this is a non-hydrolyzable analog of GTP is there. GTP, what is there? Guanyl triphosphate, okay, guanine cytosine, you know. So guanyl triphosphate, just like ATP, it is a GTP. So ATP is adenine triphosphate, GTP is a guanyl triphosphate. So this GPP is what? That is its analog, but it is not hydrolyzed. So if its production increases, and it will not be hydrolyzed, so you can count it in proper way, and on that basis you can predict that the drug is effective or not effective. Okay. So these are what various models which are available for in vivo and in vitro testing. Okay. What is now homework is, please go through the models, that is testing machines actually. What are these testing machine? As I told you, you can note it down. What are these? We have seen it earlier in the beginning. We have seen various models in that. Okay.
Pole climbing.
Yes, that's again I'll give you the list. Yes. So see, inhibitory passive avoidance, it is a pole climbing machine. Okay, these are all step down, step through, these are the machines for that. Now here this memory impairment by basal forebrain lesions in the rat, this is a surgical model. You need not to go for video for that, because this is very simple. You will understand that basal forebrain lesions will be created. Lesion means injury will be created. Then ischemia will be there in the gerbils. Gerbils are one more animals are there. Scopolamine, that is what is scopolamine? Scopolamine is
cholinesterase.
it is cholinesterase inhibitor. Yes. Anticholinergic. So very clear, this is very clear. If increase in the cholinergic activity, because we block the cholinesterase enzyme, so we get the increase in the cholinergic activity. So when there is a increase in the cholinergic activity, that causes increase in the memory. So what scopolamine will cause?
Impair the memory.
Impairment of the memory. Okay, so we can study our drug when the memory is impaired. Okay, then MPTP is the drug actually used for Parkinsonism. Okay, that can selectively damage the area. Okay, so that is one more model which can be studied, and these are shuttle box and all that can also be available for various studies. Okay, please go through this. Okay.
Yes.